In this animation, we want to show the human immune system with images never seen before. We will learn how macrophages as well as other cells and mechanisms protect our organism from death every day. Our skin, like the bark of a tree, represents valuable protection. However, the skin is also home to countless microorganisms. Among other things, they are nourished and protected by an oily fluid called sebum. Sebum is produced by sebaceous glands. Sebum also contains antimicrobial elements that can kill microbes. If they are harmful or too many microbes on the skin, the sebum is composed in such a way that these microbes are killed. This creates a harmonious coexistence between skin cells and microbes, a kind of symbiosis. These microbes also keep one another in balance and kill pathogens that can be dangerous to our cells. The protective layer of hollow organs, such as the nose, mouth, and trachea, is a mucous membrane. Mucous membranes possess cilia. Cilia move back and forth in a wave-like manner. This allows them to move mucus, to which foreign substances adhere, upward into the oral cavity. If mucociliary clearance is not sufficient, an attempt is made to achieve clearance of unwanted particles through coughing. This is done by inhalation followed by a sudden exhalation, so that foreign substances are removed from the lungs and trachea. The bone marrow has stem cells. It is the birthplace for many different immune cells, which we also call leukocytes. Stem cells divide to form daughter cells, asymmetric or symmetric. Symmetric cell division results in either two stem cells or two progenitor cells, as seen here. The progenitor cells divide again and again. Thus, a single stem cell becomes many immune cells or leukocytes. A common immune cell is the neutrophil. Some of the neutrophils remain in the bone marrow, while others make their way into the bloodstream. Our neutrophil belongs to the so-called innate immune system and moves through the blood vessels. During inflammatory processes, the endothelial cells of the blood vessels have special receptors. Thus, initially there is weak adhesion, then strong adhesion, and finally the granulocyte goes through the endothelium. This process is called diapedesis. The path through the connective tissue has already been made by other neutrophils. Our neutrophil only has to follow cytokines, that is, signal substances of other cells, a process called chemotaxis. There they are, bacteria, everywhere. They produce toxins and thus harm the cells of the organism. A neutrophil's job is to eat these bacteria. This eating process is called phagocytosis. In addition, some of the neutrophils produce certain cytokines that attract other granulocytes. other cells of the innate immune system or macrophages. They arrive on the scene before neutrophils, phagocytose bacteria, and produce a signal substance, tumor necrosis factor. It suppresses the feeling of hunger, causes fever, stimulates leukocyte production. Another leukocyte type is the T cell. T cells possess receptors. What do they need them for? Macrophages or professional antigen-presenting cells. They have the ability to present antigens via certain proteins on their surface, and this initiates certain processes. Some T cells can use their receptors to recognize exactly those antigens of the macrophages for which they have been trained in the thymus. Let us take a closer look at this. T cells are also formed in the bone marrow. They then migrate via the bloodstream to the thymus. Because they mature in the thymus, they are called T for thymus cells. In the thymus, which is one of the primary lymphoid organs, T cells form antigen receptors. Afterwards, they usually migrate to secondary lymphoid organs, such as the lymph nodes and the spleen. In the lymphoid organs, naive T cells and dendritic cells meet. The dendritic cells have already had contact with a pathogen and present the antigen on the surface. They are also professional antigen-presenting cells. The T cell scans the dendritic cell for the antigen for which the T cell was trained in the thymus. If it finds the antigen, T cell activation begins and, among other things, clonal expansion occurs. The cell reproduces itself thousands of times. These cells respond to a specific pathogen or antigen. Then, some T cells start their journey and migrate to other tissues. Once there, the cytotoxic T cells scan other cells with their receptors to look for cells that have the specific antigen on their surface. If they find their antigen in a cell, they know that a pathogen, for example, is in the cell. The cytotoxic T cells begin to induce apoptosis. The cell's debris are eventually eaten by macrophages and new cells form. There are not only cytotoxic T cells, but others as well. T helper cells, memory T cells, regulatory T cells, and others. T helper cells work hand in hand with B cells. B cells possess receptors like T cells. Like T cells, B cells also originate in the bone marrow. They then search for their specific antigen in the lymphatic system or in the bloodstream. If the B cell finds its antigen with its receptors, either activation occurs immediately or the B cell moves into the lymphoid tissue where T helper cells wait. T helper cells, as we've seen before, were also trained for one specific antigen. For this reason, the B cell presents its antigen on the surface. This allows the T cell, with its receptors, to scan the B cell. If it is the antigen for which the T cell has been trained, it gives the starting signal for the division and maturation of the B cell. As with T cells, clonal expansion occurs. Thus, thousands of new B cells or plasmablasts and plasma cells are generated from a single B cell. Mammary B cells are also produced, which will spring into action in the event of another subsequent infection. During acute infection, plasmablasts and plasma cells immediately begin to produce antibodies, and they release these antibodies into the bloodstream. Antibodies are also often called immunoglobulins. If the toxin, the bacterium, the antigen is in the blood, the antibody just produced by the plasma cell can bind to it. In this way, for example, a toxin can be rendered harmless and an antigen can be marked so that macrophages can recognize it more easily and phagocytize it more quickly. This process is called opsonization. All immune cells have a stem cell as their origin. It develops into progenitor cells and eventually into the immune cells just shown. Other important immune cells are natural killer cells and platelets. Platelets, also known as thrombocytes, originate in the bone marrow from megakaryocytes. Megakaryocytes have pseudopodia that extend into blood vessels of the bone marrow. It is through these pseudopodia that platelets are emitted into circulation. One megakaryocyte can produce 1,000 to 3,000 platelets. This process is called thrombopoiesis. The platelets move through the blood vessels of the body. If injury to a blood vessel occurs, hemostasis begins. The platelets adhere first to the collagen fibers of the connective tissue and eventually adhere to one another. This results in a quite fragile platelet plug, which prevents the blood from escaping. Eventually, fibrins transform on the platelets, which ensures a stronger plug. And it will bind more platelets and red blood cells. Thus, a strong clot is formed.